So it gives me great pleasure to introduce, yes, you can, Dr. Jack Szostak, who comes to us from Harvard, Mass General in particular, where he's a professor of genetics and also a professor of chemistry and chemical biology. He is an HHMI investigator and a member of the National Academy and has won many other awards. Notably for us here at Jack's, Dr. Szostak was a alumnus of our long-standing summer student program back a few years ago. And we would like to take credit, of course, for all of the wonderful findings that have come out of students who studied with us or did internships during the summer. And Dr. Szostak did his internship while he was a freshman at McGill University. And according to my conversation with him over dinner last night, I think we can really take credit for his findings because he was convinced after his summer in Bar Harbor that there was no way he was going to work on mice. So he went on to do, of course, wonderful work in discovery of telomeres and the sequences, and his work showing telomeres are important in aging. And this led to the Lasker Prize in 2006 and the Nobel Prize in Physiology or Medicine in 2009. And currently, then there was a major shift in Dr. Szostak's research, which is now, I think we can say, the ultimate discovery-based research, where he's working on isolating self-replicating functional biomolecules and informing us of possible origins of life. Thank you.
I suppose I should say thank you for letting me come back despite the conclusion of my summer work. It's hard to believe that was 45 years ago. But it was a really tremendous introduction to science, and really the first time I got to do really cool, interesting research projects. So that was terrific. I was incredibly impressed when I went back to the Jackson Labs in Bar Harbor a few years ago and saw what looked like a completely different campus. I mean, the development over that period of time is astounding. And during that visit, I think was the first time I heard the very beginnings of the idea of this institute. And so it's really been fantastic to come here and see that it's a reality. It's really, it's an amazing building, amazing people, and I'm sure great things are going to come from it. Okay, so since I don't work on mice or humans, I thought what I would do is give a talk that's a little bit different. That's going to be kind of half history, and people, even yesterday, people were asking me, how did I get into the whole telomere thing, and then even more puzzling, why did I get out of it? So I think I'll go through some of that history and try to explain some of the reasons, and basically it comes down to the fact that there's just so many interesting things out there, right? And during the course of a career, you can work on lots of different interesting things, and there's just a tremendous amount of stuff waiting to be discovered. Okay, so, and then I'll talk a little bit about, just give you a few snapshots of some of the stuff that we're focusing on now in my lab. Okay, so the way that the whole, my interest in telomere biochemistry got started actually came about in a little bit of a strange way, and it's basically because when I set up my lab as a new assistant professor back in '79, I was thinking about double-strand break repair. This actually came out of collaboration with Rod Rothstein. We were postdocs together in Ray Wu's lab. And a lot of that work was done by Terry Orweaver. And the basic observation was that if you introduce a circular plasmid into yeast, and it has a little bit of genomic yeast DNA on it, you'll get a few transformants. They're rare. They're the results of rare spontaneous recombination events that result in the integration of the plasmid into the chromosome. And the surprising and very counterintuitive thing that we observed and studied was that if you actually make a break in the plasmid DNA, if that break is in that region of homology, there's a huge stimulation in the recovery of transformants. And so that's basically because the predominant repair pathway for double-strand breaks in yeast is a recombination, a homology-based recombination repair pathway. If the break is in a non-homologous region, then you see non-homologous end joining, but that is normally in yeast at a much lower frequency. Okay, so these are the sorts of things I was thinking of as a new professor. You know, all the kinds of enzymatic activities that went into both repairing double-strand breaks and also, amazingly, initiating recombination during meiosis. So yeast cells, and actually many organisms during meiosis, on purpose chop their genomes up into lots of little pieces and use this repair pathway to induce recombination. So the repair pathway is, you know, it's a little complicated. There's a lot of enzymes involved in resection, various endonucleases, exonucleases. There are strand invasion and repair synthesis processes, branch migration, holiday junction resolution. So, you know, this was all the kind of stuff we were thinking about and talking about in the lab. Basically, what do cells do when they see a DNA end? Okay? And it never really crossed my mind that, of course, there are plenty of DNA ends in normal cells all the time, right? Until I went to a Gordon conference and I heard a fantastic talk by Liz Blackburn, who is working on this really bizarre organism, Tetrahymena, grows in shallow ponds. And this organism does a lot of weird things, but amongst those weird things is it takes its sort of normal micronucleus with regular chromosomes, chops them up into thousands of little pieces, and differentially amplifies them in the macronucleus. And so at a time, basically, when cloning was new and PCR had not yet been invented, Liz was able to take the ribosomal DNA from the tetrahymena macronucleus, because it's just naturally amplified, and study it, and in fact actually sequence the ends of that linear DNA molecule. And of course she found this simple repeat sequence at the very ends. And so these are stable DNA ends. They don't act like double-strand breaks. They don't get repaired. They don't show non-homologous end joining. They don't get involved in recombination events. So, you know, what's going on? They seem to act like chromosomal telomeres. But the telomeres wasn't known. And I think there was quite a bit of thought that tetrahymena was just so weird and so different and so distantly related to other eukaryotes like us, that maybe this was just another weird specialization of tetrahymena. So I was really struck by her talk, because here's a DNA end that behaved completely differently, just totally opposite to the DNA breaks that we were thinking about all the time. And it was that disconnect that made me go up and talk to her. And we had a great conversation, and we thought that even though it was a long shot experiment, it would be great to take this DNA and put it into yeast and see if it showed the same properties. Could it replicate? Would it act like a telomere? Could you have linear DNA molecules replicating in yeast? Okay, so things were simpler back then. She just got some of this DNA and sent it to me in the mail. And I didn't think the experiment was going to work, so I put it in the freezer. And then after a while, I thought, well, might as well give it a try. And oh yeah, so before I go on, I can't help but mention the fact that this piece of DNA, this ribosomal DNA from tetrahymid, is really pretty amazing. So Liz, of course, and then I studied this bit right at the end. But at just a couple of kilobases in from the end, there's an intron. And that is the intron that Tom Cech studied because, again, this is a very high copy number. He thought this is a great system. We'll find all the protein machinery that takes care of splicing, right? And it's purified, and basically the RNA does it all by itself. So that led to the discovery of ribozymes. And so 2 Nobel Prizes from 2 kilobases of DNA in one weird organism. Okay, so the experiment that I did with the DNA that Liz sent me is shown here. It's an incredibly simple experiment. We had a plasmid, had a replication origin, a gene that we could select for, cut it with a restriction enzyme, ligate the tetrahymid telomeres onto each end, take that ligated assembled DNA here, transform that into yeast, and ask, do these molecules replicate as linear molecules, which would say that these sequences are acting like telomeres? Or if they don't, we thought, well, they'll either get degraded and we'll get nothing, or they'll get ligated to each other, non-homologous end joining, we'll get circles out. And so I was actually telling someone who was here yesterday that this is, I think, the only time in my life where I've had an experiment where the result was so clear-cut and so dramatic. In those old days, we would run gels and expose X-ray film and put it into this automatic developer, right? And then the X-ray film would get developed, it would come out the other side. And as the film was coming out of the machine, I could see the bands that were diagnostic of this molecule replicating as a linear piece of DNA. So it was obvious right from that moment that these sequences were functional in yeast. So the implication of that was that the underlying biochemical machinery that's responsible for the properties of telomeres is very highly conserved in eukaryotes. And so that was a pretty exciting moment, and it led to a whole series of follow-on experiments. And of course, the key one is, well, now we know the sequence here, but how does it actually work? Okay, so at this point I should explain that there are 2 separate issues with telomeres, the 2 separate problems, both of which had been understood for decades. So one is that normal broken DNA ends are highly reactive in cells. Cells don't like them, they'll do anything they can to get rid of them, degrade them, join them together. This goes back to beautiful experiments like Barbara McClintock's studies on breakage fusion bridge cycles. The other problem, of course, is incomplete replication of DNA. And so as soon as the basic properties of DNA polymerases were worked out and the need for recognized, it was clear that at the very end of a duplex DNA, normal DNA polymerases could not fully replicate the end. There would always be some part of the lagging strand that wasn't fully replicated. Okay, and so the problem that we ended up solving really is this issue of incomplete replication. In other words, why generation after generation don't our chromosomes get shorter and shorter and shorter until there's nothing left? Okay, so we knew there had to be something going on, but it wasn't really clear what it was. Okay, so now I mentioned that most of my lab actually was working on recombination. Okay? And so it was natural for me to think about recombination-based models for telomere function. I, in fact, really wanted the mechanism to involve recombination. I thought that would be very elegant. It would tie everything together. I had all these beautiful models for how it could work. So things like unequal cystochromatid exchange, right? You can imagine adding extra DNA this way. You could do strand invasion, right? So exactly the same enzymology involved in double-strand break repair you could potentially theoretically take advantage of to extend 3' ends and add extra DNA that way. My favorite model actually is shown here, which is, well, maybe we thought chromosome ends were closed in a hairpin loop. And Liz actually had some tentative experimental evidence that suggested this might be the case. So replication would give you this kind of structure, which is isomeric with this. And this is a Holiday junction. You resolve it, and you're right back where you started with, right? Very elegant. And the only problem was that these models are all completely wrong, and they were killed by a fact, an observation that came out of sequencing. Okay? And so it was actually sequencing the DNA, the tetrahymided DNA, after generations of replication in yeast that told us what was really going on. Okay? So here's the molecule that went in, our vector with tetrahymided telomeres. We put it into yeast, and when we isolated the DNA, we actually noticed right away there was something really weird, which was that it was bigger. The ends had grown longer. Right? We didn't understand what was going on. Could have been a recombination-based property. But as soon as we were able to get hold of this DNA and get enough to sequence it, we saw that what had happened was that there was this extra DNA had been added onto the ends, but it was not the same sequence as the tetrahymena repeats. Okay? And this messier sequence, also GT-rich, but more irregular, turned out to be exactly the same as the sequence that's found at the ends of normal yeast chromosomes. Okay? So this is the yeast telomere sequence. Okay? And so obviously this could not be generated by any recombinational pathway from this. And so that led us to hypothesize in this paper that there would be a special enzyme that would add, that would synthesize extra repeats at telomeres, and that the sequence would be different from species to species depending on the properties of that enzyme. And of course, that enzyme was discovered biochemically the very next year by Carol Breiter, who was then a graduate student in Liz's lab. Okay, so over the years, many people have worked on this enzyme, telomerase, and it's now known to actually be a ribonucleoprotein, and the RNA component, among other things, acts as the template that determines the sequence of the repeats that are added on to the ends by extending the 3' end of the telomere DNA. The protein component, or one of the protein components, is a reverse transcriptase-like enzyme that actually does the DNA synthesis. And there's a lot of extra RNA, which is extremely variable between species, and which seems to act as a scaffold to hold on to a whole bunch of accessory proteins that take care of various jobs, including interaction with proteins that are localized to cellular telomeres. Okay, so this is one of the last experiments that we did in the field of telomere biology in my lab. This is work done by Vicki Lundblad, and she was a postdoc. And Vicki decided to go after mutations that would affect the ability of the cell to repair and build new telomeres. And so the first one that she found was called Est1, for ever shorter telomeres. Turned out to be a component of telomerase. And in the mutant, you can see here that so this is increasing generations, the telomere bands get shorter and shorter. Okay. But the interesting part of this is the biological phenotype, and that is a delayed senescence phenotype. So this mutant, after 25 generations, the colonies look almost normal. You can barely tell there's anything going on there. After around 50 generations, you can see they're starting to look a little raggedy. 60 to 70 generations, the colonies are much smaller, and by around 90 generations, you can barely grow them at all. There's a tremendous amount of cell death going on. There's chromosome loss. There are chromosome aberrations. And this all makes sense in terms of the cellular telomeres getting shorter and shorter. Eventually, you get into some essential region and the cells die, or else you get short enough that the normal cellular machinery that stabilizes the ends and prevents them from undergoing ligation reactions is missing on at least one or a few chromosomes, and so you start to get end joining and then chromosome breakage and the whole panoply of chromosome aberrations. Okay, so so that was a pretty exciting result, and in Liz's lab at about the same time, they found very similar results from different kinds of experiments in tetrahymena. And so this made a kind of pretty obvious connection to aging. As cells go through a certain number of generations, basically if they don't have enough telomerase, their telomeres get shorter and things start to go south. And it also led to a very simple, and as I'll explain in a minute, a very incomplete model for the initiation of tumors. And basically the idea is that if cells don't have telomerase, they have a limited lifespan in terms of number of generations, number of divisions they can undergo. But if they do have high levels of telomerase, then they're essentially immortal. They can divide an unlimited number of times, and of course that's the hallmark of cancer. And very soon after, it turned out that a very, very high fraction of tumors turn telomerase back on. So it's normally off or in very low levels in most of our cells. But many, around 90% of tumors turn telomerase back on. Okay, so this is kind of the original simple model. So of course, nothing's ever that simple in real life. And a lot of the, I think the insight into the greater complexity came from studies by many people, but especially by Tizia DeLange at the Rockefeller, on the connection between double-strand break repair and the repair signaling pathways, the connections between those pathways and telomere biology. And I don't have time to go into all of that biochemistry, which is really very beautiful and elegant and very complicated. But the basic point is that normal double-strand breaks allow for a variety of repair pathways, but they also induce damage signaling pathways, which can lead to cell death. Normally, telomeres are protected. They don't induce these repair pathways, and they also don't engage they don't induce the signaling pathways or the repair pathways. But as they get eroded down shorter lengths, then this process starts to break down. Okay, so the implication is that in our cells, when there's basically stem cells are getting towards the end of their natural life, or there's some hyperplasia, telomerase levels are low, you have some telomere erosion. Right? Telomeres are getting shorter and shorter. And at some point, you're going to induce a DNA damage response when you have essentially unprotected ends. Okay, so there's a safety net, there's a checkpoint. And so if the DNA damage response is induced and all of the signaling components like p53 are intact, then everything's fine because the cell cycle arrests, the cell dies, no harm done, except for some dead cells. If that does not happen, so if you have, say, mutation in p53 or other DNA damage response components, cell division continues. Okay? And so this is bad because now you have unprotected telomeres which are starting to get joined together and induce chromosome abnormalities and dicentric chromosomes, broken chromosomes, etc. And so that can act as a trigger for this incredible spectrum of genomic rearrangements that we see so commonly in tumors. Now, again, there's another safety valve, right? So normally, telomerase levels are quite low. And if they stay low, again, it's no problem. The cells are going to die eventually, no tumor. But when telomerase gets reactivated, then the cells can continue to grow, and then you get a tumor. So in the early stages, it's the low levels of telomerase that are a problem, and in the later stages, it's the high levels that are a problem. Okay, so it's a bit more complicated than we thought originally, but pretty interesting developments to watch for me from a distance over the years. Okay, so people often ask me, so why did you leave this field and how did you get interested in things like evolution and the origin of life? And basically, it's been a long, winding path, but as you've heard, I didn't really want to get into doing mouse experiments. There's just some incompatibility there. I just don't like cutting them up, I guess. And I also felt like, and I was correct, in that a lot of really brilliant people were going to come into the field and do the experiments that needed to be done. So I thought this is a good opportunity to kind of look around and think about maybe doing something else. And so I actually sat in on some courses at Harvard on neurobiology, which back then just seemed like too hard. There just wasn't the technology to do the things that, of course, now we can do. I looked into a bunch of different fields and eventually settled on what I thought was quite a conservative way to switch fields, which is to get into RNA biochemistry. It's basically the same kind of molecular biology we've been doing. It works, it Altman's works, sort of fresh and new. I thought there were a lot of interesting questions. And surprisingly, not a lot of people going into that field. So for about 5 years, I started to sort of bring more RNA biochemistry into the lab as we phased out the yeast genetics. And we started off, of course, working on the ribozymes that nature has given us. And there's really not that many of them. We were trying to force them to do replication-related chemistry because as soon as ribozymes were discovered, everybody jumped to the conclusion that life had to begin with self-replicating RNA. So we thought, well, let's see if we can make a self-replicating RNA. Trying to do that with natural ribozymes that still exist, really, I think we pushed it as far as we could, and it didn't go very far. So that made me realize that what we needed to do was to be able to evolve our own ribozymes that did what we wanted. So through the work of a lot of great people in the lab, and so initially Andy Ellington, we worked out ways of evolving populations of molecules. And I should say at pretty much the same time, Jerry Joyce at Scripps was doing similar work, and Larry Gold, UC Boulder was doing the same kind of thing. And basically we all basically came to the same technology, which is to begin by making a huge library of essentially random sequences. I think this is sort of the ultimate metagenome, right? Everything is possible in this initial random library. We apply a selective pressure, and so you have a tube of molecules. You can apply any pressure that you want to filter out the ones that don't do what you're interested in and enrich for the ones that do. Then since there aren't that many of those, you amplify them, and you can introduce more mutations if you want. And you can go around this cycle of selection and amplification. And so that's basically, encapsulates all the ideas of Darwinian evolution right there. You have heritable variation, you have natural selection, except it's not natural. In this case, it's experimenter-imposed selection. And eventually, the structure of the population changes. And if you're lucky and things work out, the population gets taken over by a molecule that does what you want it to do. And then you go to sequencing, and then you're back into doing messy biochemistry to figure out what actually happened. And so I'm not going to go through a lot of that, just show you a couple of snapshots. This is one of the first interesting RNA aptamers that we came up with. It's just a little RNA NMR structure shown here. It has a little sort of pocket on the surface that is complementary in shape and electrostatically to ATP, which it binds very selectively. And so it's turned out over many experiments. So you can basically take random sequence RNA and pretty easily find sequences that will bind just about any target molecule. So at the time, I think that was kind of surprising. People, I think we all thought maybe you need the functional complexity of proteins to do that. But It turns out that RNA, despite its limited diversity of building blocks, is really quite capable. In fact, we were able to isolate ribozymes, and through work of Dave Bartel, we got this ligase ribozyme. It does a template-directed ligation reaction, and people are actually still working on this ribozyme and evolving it, and it's been evolved at this point to an RNA polymerase ribozyme, an RNA molecule that is an RNA polymerase that can, on a restricted range of templates, can make molecules longer than itself. The holy grail of this effort is an RNA that could replicate its own sequence, but that's still a ways away. Okay. In the mid-90s, we got into trying to extend this to proteins, and so what you see here is a little ATP binding protein. It looks just like any small protein domain that you would see in biology, but it's completely non-biological. It was evolved from a random set of protein sequences. And it has a little pocket on the side that binds ATP. So I think these experiments together told us, among other things, that it's really not that hard to get function out of collections of random sequences. In both the RNA case and the protein case, these functional domains with very good affinities and specificities are present in the random sequence pools at frequencies on the order of 10 to the minus 10th, which sounds low, but in terms of actual mass of molecules, it's not much. Okay, so after working on that kind of stuff for about a decade, I started to get increasingly curious about how things got started in nature. Okay, it's actually very easy to do this kind of evolution in the lab, but it seemed really hard to imagine how things got started on the early Earth. Okay, and so this is the kind of the way that we're thinking about it. And we've been thinking about the origin of life. Since all life that we have is cells, we're thinking about the origin of cells, right? And so the idea is you have some kind of primitive cell membrane, a self-assembling structure made from molecules that would have been available, that in some mysterious way can grow and divide without any biochemical machinery, just in response to environmental conditions or fluctuations. And inside that, some kind of informational molecule, possibly, maybe even probably RNA or something like RNA, that could replicate, again, without enzymes, in a purely chemical process, that would allow for the transmission of information from generation to generation, but would also allow for sequences that could do something useful. And so what we're hoping is to be able to build structures like this and take them through cycles of growth and division. And by starting with a bunch of random sequences, maybe find sequences that confer a selective advantage on their host cell and start to take over the population. That, I think, would be the beginnings of Darwinian evolution. And that's what we'd like to see. And of course, we'd like to know what the first functions that arise under these kinds of conditions might have been. Okay, so the membranes, as I said, are self-assembling structures that can easily be generated by simple fatty acids. They make these beautiful structures. And it turns out that actually making them grow and divide is not that hard. We can do a cycle of growth where we add new food. They grow in this unusual way into filaments that make the filaments are very fragile and divide easily in a number of different ways. And so we have a cycle of growth and division that can be repeated indefinitely. The harder part is the RNA, and that's what we're focusing on mostly these days. There's a whole laundry list of problems that have kind of stalled progress in the non-enzymatic replication of RNA. This is something that people have been working on for 50 or 60 years. A lot of progress was made early on by Leslie Orgel and Jerry Joyce and others, but it kind of stalled because of all of these different problems. And so we've come back to kind of look at these again, and I think I'm running out of time, so I'll just go very briefly over a couple of ideas. One of the problems with RNA is that you make these incorrect 2' linkages. And we've shown that that actually doesn't matter that much. You can still get function even with a very heterogeneous backbone. And the reason is that the structural perturbation, like the different sugar conformation here, is really localized to the site of that structural heterogeneity. If you look at the overall duplex structure here, it almost looks like a normal little piece of RNA. And the variation at the site of this heterogeneity is actually compensated for by variation in the flanking nucleotides. So what we thought was a huge problem actually turns out to be not a problem at all. Let me just show you, this is kind of the state of the art at the moment, where we're able to combine limited chemically driven template copying and actually start to copy sequences inside these vesicles, which can be replicating structures themselves. So we can now encapsulate primer template sequences. We can add nucleotides, which will spontaneously cross the membrane. The chemistry requires magnesium, which would normally destroy the membrane, but when it's complexed with citrate, interestingly, the membranes are protected and the RNA chemistry can still go on. So now we can do primer extension template copying inside these vesicles. So the hope is over the next few years, if we can solve some of the remaining problems, we'll be able to do complete cycles of RNA replication without enzymes inside vesicles, which are themselves replicating structures. And at that point, I think we'll be able to just kind of turn the crank, go through generations, and hope to see the evolution of novel functions. Okay, so this is our idea of an environmentally driven cell cycle. So in a geothermally active region of the early Earth, something like Yellowstone Lake today, you have essentially hot springs, plumes of hot water. Protocells get caught up in these. They get transiently heated, which drives strand separation, and then immediately quenched in the surrounding cold water, which gives you time to copy the separated strands. And the heating also allows for a rapid influx of nutrients that are made in the environment, and again allows for growth and template copying. So this is essentially an environmentally driven cell cycle. And this is, I think, the most plausible kind of natural environment that could support the kind of chemistry that we're looking at in the lab. Okay, and all of this, of course, has been done by a tremendous number of talented students and postdocs. And so this is a picture from about 5 years ago. So I'm grateful to all of them, and thank you for listening.